谢谢郭师兄的邀请和介绍。很高兴今天给大家分享我的工作。然后，在演讲过程当中，如果大家有任何问题，可以随时打断我，用中文和英文都可以。然后今天要分享的工作是一个在基底神经节当中，纹状体的一群直接通路的神经元介导负向的强化学习。The basal ganglia plays a very important role in motor control and reinforcement learning. The dysfunction of basal ganglia can lead to various motor deficits such as Parkinson's disease, Huntington disease, or obsessive compulsive disorders. The neurodynamics of various basal ganglia nucleus correlate with reinforcement learning, and the lesion of them could impair the reinforcement learning. Here is a classical model of the basal ganglia. Striatum, the major input of the basal ganglia, it receives input from the cerebral cortex. It sends direct and indirect projection to the output nucleus of the basal ganglia, SNR GPI, forming the so-called direct and indirect pathway. The direct pathway spiny projection neuron mainly express DRD1 receptor, and the indirect pathway spiny projection neuron mainly express DRD2 receptor. Here is the classical views of the basal ganglia. Activation of D1 neuron promotes movement, while activation of D2 neuron suppress movement. Activation of D1 neuron drives reward, while activation of D2 neuron drives aversion. This kind of classical views provide a very simple but elegant explanation for the control of movement and also the learning of bad and good. Besides the classification of the striatum into direct and indirect projection neuron, the striatum can also be divided into distinct compartments based on the expression of neurotransmitter-related molecular, the striatum and the matrix, such as the striatum is kind of a patch-like structure in the striatum. The striatum is very large, and the striatum is kind of distributed in the striatum. And specifically, the striatum are usually identified by the enriched mu-opioid receptors. Previous studies have suggested that the striatum plays a very important role in a lot of different learning behaviors, such as motor learning, reward-guided learning, cost-benefit decision-making, or reward prediction. But the role of the striatum in reinforcement learning has rarely been causally tested due to the lack of the transgenic mouse line to specifically label the striatum and then monitor the neural activity. So our first motivation is to find a genetic marker that can specifically label the striatum. Here is the LM atlas, brain atlas for the t-shirt, and we can see that the t-shirt mRNA forms this kind of cluster structure in the dorsal striatum. And this kind of structure resembles the structure of striatum. And coincidentally, this t-shirt has been reported, identified by the single cell sequencing in recent two papers. Another genetic marker we used in our paper is the published genetic marker prodanophin. And to confirm that indeed the prodanophin in label a striatum neuron, we crossed the prodanophin cream mouse with the AI14 mouse. And we found that the neuron in the dorsal striatum, they have this kind of clustered structure. It is patch-like structure. It's also likely to be the striatum neuron. First, to visualize the expression of the t-shirt neuron, we crossed the t-shirt flip mouse line with the reporter mouse line. And this is a sagittal section of the t-shirt TD tomato mouse. And this is the dorsal striatum. And we can see that the T-shaped neuron in the dorsal striatum may form this kind of cluster. And to further confirm whether this neuron really belong to the striosome, so we did the immunostaining of the mu-opioid receptor (MOR) to see whether the T-shaped neuron are indeed located in the striosome. And indeed, we found that the T-shaped neuron cell density in the striosome is much higher than the surrounding matrix. And to explore the expression profile of the T-shaped in relationship to the DRD1 or DRD2, we performed the RNA-scope of T-shaped together with D1 and D2, and we found that T-shaped neuron mainly express DRD1 but not DRD2. This result shows that the T-shaped define a population of striosome neuron, and those neurons belong to the direct pathway projection neuron.
你好，不好意思，可以打断一下，我想问一下，就是你这个 marker 只标记纹状小体的神经元吗？就是其他脑区、其他神经元都不标记吗？
它标记除了纹状小体的神经元，还标记非常多其他地方的神经元，比如说这边的 nuclear combus， 还有其他地方我没有秀出来的，比如说 olfactory bulb， 还有一些 centromeres 的地方，还有特别是小脑当中也有很多的标记，就它标的 neuron 还是很多的，不仅仅是 striosome。
有神经元特异性吗？是椎体中间神经元，就是指标中间神经元。
你是指在 striatum 当中吗？
全脑。
全脑的话，我们没有认真看它具体标记哪种类型的神经元，是中间神经元还是其他神经元。但是在 striatum 当中的话，就是它标记的并不是中间神经元，它主要标记的是就是 projection neuron。
这个 projection neuron 是椎体神经元吗？
它不是椎体神经元，在 striatum 当中就是它比较特殊。striatum 当中的中间神经元一般主要有3类。中间神经元的话，主要有 chat， 然后 PV， 然后 SOM， 然后我们做了染色，就是发现 T-shaped 的，就是并跟它们都不 overlap， 就是它并不是标记的是 interneuron。
挺有意思的，好，谢谢啊，不好意思。
Okay. Next, we want to determine whether T-shaped and prodanophin label the same population or different population in the striatum. So we perform the RNA scope of the T-shaped together with prodanophin and also DRD1. We found that, the video covered the right side of my slides. Yeah. Let me change a little bit. Okay. So we can see that the T-shaped and prodanophin, they co-localize the percentage is very, very low. And the T-shaped neuron, a lot of T-shaped neuron co-express DRD1, and also this is true for prodanophin neuron. And 88% of the prodanophin neuron, they express DRD1. But only 2.8% of the prodanophin neuron, they express T-shaped. So basically, T-shaped and prodanophin define the two separate population of neuron in the striatum, and both of them label the striosome and direct pathway projection neuron. First, the obvious question to ask is what downstream targets do those neurons project to? So we perform the anterior grid tracing for the T-shaped and the prodanophin neuron. In the T-shaped flip and the D1 cray mouse we simultaneously injected the flip-dependent EYLP and the crate-dependent M-cherry. So this enabling us to express the EYLP in the T-shirt neuron and M-cherry in the D1 neuron. And we injected this virus in the dorsal stratum, and we can see that they project to multiple downstream targets, GTE, GPI, SNR, SNC. And when we compare the projection pattern of the D1 neuron and the T-shirt neuron, the projection pattern is very similar. So basically, the T-shirt neuron send a direct pathway projection pattern. And we also use the anterior treatment for the prodanophin, and we can also see that the projection pattern is very similar as the T-shirt and the D1 neurons. To test what kind of valencies do this neuron drive, we first expressed the channelopsin either in the T-shirt neuron or prodanophin neuron and tested these mice in a real-time place preference test. In this test, so the laser stimulation was turned on when the mouse was stay in one chamber. For example, in the left stimulation block, the laser was turned on when mice were staying in the left chamber. And we can see that the T-shirt flip mouse, they spend very less time in the stimulated chamber. But as a control for the prodanophin cray mice, the prodanophin cray mice, they like to stay in the chamber paired with the laser stimulation. So basically, the T-shirt neuron, the activation of T-shirt neuron kind of drives the mice avoid the laser stimulation, but the stimulation of the prodanophin drives the animal to receive the laser stimulation, to stay in the stimulated chamber. And to further ask whether this stimulation could reinforce animals' behavior, we tested the T-shirt flip mice in a choice stimulation test. In this test, so one side of the low smoking was paired with the water reward plus the laser stimulation, and another side with water. And during the test, the laser stimulation kind of biased away the mouse choice to get the laser stimulation, indicating in this T-shirt neuron activation kind of drives the elective reinforcement signal to the mice. And for the prodanophin cray mice, we tested them in a self-stimulation test. In this test, one side, for example, in the left side was paired with laser stimulation, and another side paired with nothing. And we found that the mouse would crazily poke the side to receive the laser stimulation, even though they didn't get anything, just the laser stimulation. Indicated this results show that the prodanophin activation, they can provide this kind of positive reinforcement signal. So this result is kind of very interesting. So previous results show that the direct projection neuron only drives the reward, but we found a different population of the direct projection neuron. They drive different valencies. So we want to ask, how does activation of T-shirt neuron drive aversion? And although the majority of the T-shirt neuron, they express DRD1, still there is a small fraction of the T-shirt neuron, they express DRD2. So there's two possibilities to explain why T-shirt neuron drive aversion. So the first possibility is that the activation of the D2 positive and T-shirt positive neuron dominated the behavior output. So we observed the aversive responses by activation of the T-shirt neuron. Another possibility is that the activation of D1 positive and T-shirt positive neuron indeed they induced aversion. To resolve this, we we applied the intersect virus strategy. We by combination of the flip and the cray in combination with the tissue flip, D1 cray or A2A cray mice to only target some kind of specific component of one population of neurons. First, we confirmed the previous results. We stimulated in the A2A cray mouse. We injected the cray-dependent channelopsin, and we can stimulate the D2 neuron. And indeed, we found the stimulation made the mouse avoided the laser stimulation and the drive aversion. And in the t-shirt flip D1 cray mouse, we injected the cray-on flip-off channelopsin. So this virus expresses the channelopsin in the t-shirt negative but D1 positive neuron. So this is supposed to be the traditional D1 neurons. And in this, when we activate this population neuron, the mouse would spend a lot of time in the stimulated chamber, indicating its kind of driving reward. And to specifically label the t-shirt positive and D1 positive neuron, we injected the cray-off flip-on viral channelopsin virus in the t-shirt flip and A2A cray mouse. So this virus enabling us to express channelopsin in the D2 negative but t-shirt positive neuron. D2 negative is kind of equivalent as D1 positive. So this can enabling us to express channelopsin in the t-shirt positive and D1 positive neuron, and we found that activation of them drive aversion. And another evidence to support this is that because the D2 neuron only project to GPE, but the D1 neuron, they project to the GPI and SNR. So we stimulated the terminals of the tissue neuron and the prodanophin neuron in the GPI. We found the terminal stimulation of the tissue neuron in GPI, they induced aversion, but for prodanophin, it is rewarding. So another evidence to support that indeed the activation of the specific t-shirt direct pathway projection neuron induced the aversion. So next, we want to ask what the population neuroactivity look like when animal during associative learning behavior. So we trained the mice to perform a Pavlovian conditioning task. In this task, an auditory cue was paired with water, and another auditory cue was paired with air puff as punishment. And we found that the tissue neuron, they were very highly excited by the air puff, but not the water reward. But for the prodanophin neuron, the activation, the prodanophin neuron, they were activated by both the reward and the punishment. So this result is kind of very contrasting with the previous result, and especially the t-shirt neuron, and they represent kind of negative reinforcer signal. So next, we focus on our study of the tissue neuron. To monitor the single cell neuroactivity of the tissue neuron in behaving animal, we first expressed the GCamp in the tissue neuron and monitor the neuroactivity through implanted green lens and use a one photo microscopy to observe the neuroactivity. And we found that many tissue neurons, they were excited by the air puff. And also some neurons, they were excited by the water. And spatially, they are intermingled with each other. And here shows some example neuron. And this example neuron shows that this neuron shows high activity to the air puff, but no activity to the water. And this example neuron show the opposite. They were excited by the water, but no response to LPAF. So basically, the teacher neuron exhibited heterogeneous neural activity, and as a population, we also look at the percentage of the neuron excited by the LPAF and the neuron excited by the water, and we found that there are significantly larger fraction of neuron they were excited by the LPAF, and the amplitude of the neuron responding to LPAF was also much larger than that of the water. And for the neuron, they were responsive to the punishment to the LPAF. The activity was also scaled with the LPAF duration. To look at the neural activity during the associative learning process, so we train the animal also in the Pavlovian conditioning task, and this is an example session shows the mouse show the predictive licking during the tune predicting the water, but no licking to the LPAF. And this is an example neuron that after learning, it showed this neuron showed the predictive activity to the tune of the punishment. And to visualize the neural activity of the population activity during the punishment and the reward trial, and we performed the PCA to reduce the neural activity, and the red line indicating the punishment trial and the green line indicating the reward trial. The red dot is the offset of the punishment cue, and the green dot is the offset of the reward cue. And we can see that the trajectory of the neural activity in the punishment is very different from that in the reward trials. And we also use the population activity to decode, whether to distinguish whether the CS1 versus CS2. And before learning, the neural activity could not distinguish the rewarding cue and the aversive cue, but after learning, they can distinguish it. This result indicating that the teacher neuron is kind of, they can involve in the learning process, and they eventually they will develop the predictive signals. To further test, to examine whether the teacher neuron were indeed engaged in the actively avoiding the punishment behavior. So we trained the mice to perform active avoidance task. In this task, the mouse had to run after an auditory cue. They need to run to surpass a certain threshold to avoid the LPAF. Otherwise, they will get LPAF. And the punishment trial and the neutral trial were randomly interleaved. And this is an example session showing the behavior. So in the success trials, the mouse would exhibited a higher running velocity in the response window, but for the failure trial, the mice did not run significantly higher to avoid the LPAF. And this is some example neuron, and we found some teacher neuron, they were only excited when in the failure trial when they received the punishment, and then this result is kind of similar as what we show in the photometry data, that they were reporting the perception of the punishment. And another population of neuron, they were activated when they were actively engaged in the running process. But when they were receiving the punishment, actually they were not really so activated, indicating this neuron, they were kind of only actively engages in active avoid the punishment. To further measure the neural activity of the tissue neuron during the response window and animals' running velocity, and we performed this kind of linear correlation analysis, and we found that, for example, for this example neuron, and it showed the positive correlation. When the animal had higher running velocity, the tissue neuron had higher activity, and there are many of the tissue neurons show this kind of positive correlation. And we also performed the PCA analysis to show the trajectory of the tissue neuron activity in the success trial and failure trial and neutral trial. And interesting, we found that the neural activity of the in the success trial is very different from the failure trial. And the failure trial activity is kind of more closer to the neutral trial activity. And also this kind of activity during decision window can be used to decode the animal's behavior. Last, we want to ask whether the tissue neurons are important for the reinforcement.
肖像我可以问你一个问题吗？前面那个。
好。
你在 intertrial interval 的时候，如果老鼠在运动的时候，你那些神经元对运动起不起反应？就普通的运动，不是 active avoidance。
我们看了，就是在 intertrial interval 的时候，就是这些 neuron 就是反应很少，就是基本上不参与反应。就是这群 neuron 好像就是说，它好像并不是单纯的跟运动相关，并且我们就是如果激活这群 neuron 的话，也就是没有看到就是它促进运动。Next, we used the cord, a new dread chemogenetic tool to inhibit the tissue neuron. So we trained the mice to perform an auditory go-no-go task. In the go trial, the mouse need to lick to get the water reward. Otherwise, they will not get the water reward. In the no-go trial, the mouse need to withhold the licking to avoid the LPAF. Otherwise, they will get the LPAF. So this is an example session for the EYP animal and the cord animal during the early learning stage. And we found the cord animal had much higher false alarm rate compared with the EYP animal, indicating that they have kind of impaired no-go suppression behavior. And when animal fully learned the behavior, when we apply the cell B to inhibit the tissue neural activity, the inhibition does not affect the animal's behavior. This is a summary data. So the tissue neuron inhibition did not affect the go trial, but it significantly decreased the animal's correct rejection rate. The animal had a higher false alarm rate. And the impaired animal's behavior is mainly due to the higher false alarm. And next, we want to test whether this kind of higher false alarm is kind of generally because the animal kind of have higher movement. So we also checked whether the inhibition of the tissue affected the animal's movement, and we tested them in the open field test, and we found that they did not affect the animal's running velocity or distance.
再问一下，这个抑制影响它那个 active avoidance 那个 behavior，它影响吗？那个 active avoidance 那个？
你说影响这个行为是吧？影响 active avoid the LPAF behavior。这个的话，我们没有 test 它，我们觉得可能应该是会影响，因为我们发现有很多 neuron 在这个 task 当中被激活，然后很多 neuron 确实就是跟 animal 的 这个 active 的这个 avoidance 的 vigor 就是 positive correlate， 所以我们觉得它应该如果抑制它的话，会影响它的 active avoidance， 但是我们没有做这个实验。
那这个激活它会影响它的 spontaneous locomotion 吗？类似的。
激活它的话，就是不会促进它的 spontaneous movement， 但是会减小一点点它的 movement。但是这种减少就是一种解释是，就是它可能是减少了运动，但另外一种解释就是因为这种 activation 是给了老鼠一种 negative 的一种 feeling， 因为它给了老鼠一种 negative 的drive aversion， 就是这种运动的减少就是 kind of difficult 就是去说，对。但是就是说它的激活的效果跟 prodanophin 是完全相反。然后如果你看到这个视频，就是我之前看到秀的这一个视频当中，会看到就是说激活这些 tissue neuron， 它其实并没有影响就是 mouse 就是去 avoid 这个 laser stimulation， 但是就是说 activate 这个 prodanophin neuron， 你可以看到就是很多时候就是老鼠在做一些就是 rotation 的这种运动，就是 unilateral 的就是 rotation， 就是 prodanophin neuron， 我觉得它可能就是会更多的 involve 在一些 motor 的 control 当中，但是我觉得 tissue neuron 好像就是在这方面 involve 的比较少。
那这个除了那个 placement avoidance， 这个可以通过和 Q pair 在一起，能够引起那种lective reinforcement learning 吗？
跟 Q
pair 在一起，
跟 Q pair 在一起，就是激活它，看它能不能影响它的 learning。
对，那是否能够直接形成 lective reinforcement learning？
我们没有直接做你说的这种conditioning， 就是 Pavlovian conditioning 那种，但是我们做了一种就有点类似于 operant 的这种 conditioning， 就在这个 task 当中，就是老鼠 first 它就是需要 poke 这个 central 的 port， 然后它 either 去 choose 这个 left 或者 right， 就是 one side 是 pair 的，就是一个是 water reward 是它的 size 是一样，但是其中一个是有 laser stimulation 的。然后在比如说最开始的前面20个 trial， 我们会 force 这个 mice 去 follow 这一个 Q， 就是这个 light Q， 然后在 free choice 的时候，然后我们去 test 它，看它就是 choose 哪边，就是我们发现就是老鼠更多的就是choose 就是那个 side 就是没有那个 laser stimulation 的 side， 就好像这个应该就是类似，就它其实并没有直接的影响运动，它只是影响了它的一个 choice。就我觉得这个应该也是一个间接的一个证据，说明就是说它是可以提供一个 negative 的，就是 reinforced 的信号。
对，这可以。
Okay, so this is a working model we propose for the T-shirt neuron in the circuitry of the basal ganglia. So we think that the T-shirt neuron in the striatum may provide a negative reinforcement signal to the downstream target GPI, and another population of the striatum neuron, prodanophin neuron, they provide the positive reinforcement signal to the GPI. And the GPI integrates the positive and negative reinforcement signal and send projection to the lateral cellular RMTG, and eventually send project to the SNC and VTA to influence dopamine activity to influence the learning process. To sum up, so we found a population of molecularly defined striatal DRD1 neuron drive aversion through the direct pathway projection. This is kind of very different from the traditional view. And we found that teacher neuron and prodynophin neuron in striatum, they provide positive and negative reinforcement signal to facilitate the learning. And the inhibition of teacher neuron, they impair the learning, mainly through the increased insensitivity to the negative feedback. So for the future extensions, we have two questions that want to continue. So the first question is, how does the striatum control dopamine signal to support learning? Because both the teacher and prodynophin neuron, they send projection to the GPI, and the activation of the terminal of teacher neuron in GPI, and they induce aversive responses, while activation of the prodynophin neuron in the terminals in GPI induce rewarding responses. And previously, our lab has a paper showing that specifically a population neuron in GPI sending projection to the lateral habenulum, they are involved in the action evaluation behavior. So we want to know whether the teacher or prodynophin neuron, how they will influence dopamine activity or GPH neural activity, how they will eventually influence action evaluation and learning behavior. And another question we want to ask is that whether the teacher and the prodynophin neuron, they have this kind of interaction. There is more increasing evidence showing that the projection neuron, they not only send signal to the downstream targets, actually they also have a lot of collateral interaction. So whether they have this kind of neuronal interaction locally in the striatum is also unknown. So because the t-shirt is driven by t-shirt, we have the t-shirt flip and the prodynophin gray mouse. So if we cross these two mice, so we can use a flip dependent GCAM and the flex dependent red GCAM, and we can simultaneously monitor the neural activity and to see how they will behave in the behavior. Yeah. And lastly, I would thank Bo for his support and suggestion, and also he asked a lot of reagents for me, and also Hanfei for a lot of surgery and imaging experiments, Alessandro for the iron scope, and also a lot of other lab members for the helpful discussions, and the distros and Handman lab for providing us a lot of viruses, and the Austin lab for helping us to acquire the STP images, and the Zeta lab for providing us the D1 Cray and A2A Cray mouse line, and also our long-term collaborator Josh Wang. They helped us generated the mouse line, and also Xu Yi and Yongjun, they provided us a lot of reagents and the mouse lines. Okay, thanks. Thank you for your listening.